Alrighty, so this is the first video. So week one, and for immunology and infectious diseases. And this is video one of week one. So these are very light details, an overview of what has been addressed. So as noted in the first PDF, let's take a look. As noted in the first PDF, the main topic is immunology, an overview of immunology, and a sort of introduction into innate immunity. And the second major topic is B cells and antibodies. And the third major topic is T cells. Um, and here are the 7 major subsections of the first topic: an introduction, an overview of the immune system, epithelial barriers, the complement system, cells of innate immunity, and inflammation, and innate recognition of non-self and damage. So that's 7 subtopics. For topic number 2, it focuses on B cells. The first topic is an overview of adaptive responses, and then antibody structure, followed by antigen receptor diversity, and then antibody isotopes. Finally, antibody response. It finally gets to the mechanisms, stops describing the antibodies, and it finally tells you what it does. And again, this focuses on B cells as compared to T cells. You have the following 5 subtopics: T cell receptors, T cell types, T cell activation slash antigen presentation. The antigen presentation is done by the antigen presenting cells, otherwise known as APC. And then you have information about the MHC molecules, and then the type 1 interferons. So the way that I saw fit to approach this was by going back to as far back as histology. So during histology, we addressed, well, while covering the histology of blood cells, we addressed a concept, the concept of, what's it called? Let me get there. We addressed the concept of, that's not it. Yeah, this is it. We addressed the concept of hemopoietic stem cells. And then these gave way to precursors, such as this one. So think of columns that are vertical columns, and this is how things progress in terms of differentiation of the stem cell. The differentiation is going that way, to from left to right. So we start with a hemopoietic stem cell, and then differentiation happens, and you get this variety of different blood cells. So in order to address all these topics, I saw it fit to go back to these notes that date back to histology, and work off of the same diagram that we used previously. And this is all handwritten. There is one specific slide that had this type of diagram. So I'll find out later if necessary, but for now, this is the hand drawn diagram that I did in order to just put it in a format that I think makes better sense. So let's address this flowchart of stem cell differentiation before addressing all of these topics. So, as I mentioned, you start out with a hemopoietic stem cell, and that gives rise to um, these two types of cells: a common lymphoid progenitor and a common myeloid progenitor. Those details, from what I can tell, don't really matter. It's just good to know in the back of your mind what are the precursors for the final differentiated and mature cells. What are the precursors? It helps to just have a sense of where they came from, and surely this information will become relevant later throughout the rest of med school. So anyway, you have yet another column here of more differentiation that took place, and you keep going, and eventually you're in this column right here, where this whole region is stuff that happens within the bone marrow, as you can see. So you have all these precursors. That's why the word pre is all up in here. And next, there's some more maturation that takes place in the blood, or once these certain cells reach the blood, then they have this form. They exist in this form. So down here, it covers a lot of red blood cell stuff. We're gonna ignore that for the most part. We will focus on all of this stuff, all of these, which are leukocytes. Leukocytes meaning white blood cells. So um, there's a difference of what's going on in the blood in terms of the natural killer cells, the T cells, B cells, etc., what's going on there as opposed to what's going on in connective tissue. And once you start talking about the concept of connective tissue, you begin to um, consider the following question: is the cell of interest a resident cell? Yes or no? If yes, that becomes pretty relevant in terms of immune response. So that's part of what we will get to. So just a quick overview of what are the cells that are furthest to the left in this diagram. You have NK cells, T cells, and B cells, which within the connective tissue transform into plasma cells. And these are all lymphocytes, and they have experience and development in peripheral tissues. So you also have dendritic cells, neutrophils, mast cells, eosinophils, and macrophages, which came from monocytes. And apparently, this isn't really relevant right now. Something about osteoclasts, and that's it. So now let's move on to here the actual immunology no, class notes. So here in this page, you see all of the that diagram copied and pasted. Okay, there we go. We will leave that alone. And in the next page is where we will begin to build upon it. So a lot of stuff got added to it, but bottom line here is that same diagram with, and over here you see all of the types of acronyms of not acronyms but the shorthand notation for different things that different terms that appear there in the diagram. So some of the first thing to point out is that with this purple, hot purple, neon purple color, I've circled the things, the cells that are relevant during the course, or during the class notes for week one, and it's essentially all of them. And plasma cells do get mentioned eventually in regards to memory cells or in regards to secondary immune response. So um, obviously that gets mentioned because you see the B cell got is here highlighted in that neon purple. you got these other ones that will become relevant. And essentially, it's just the mast cells which are not heavily involved or heavily addressed, at least not during week 1. So when it comes to mast cells, um, oh, this is supposed to say due to many stimuli. Okay, so the thing that was um, the material that addressed mast cells was pretty limited. It just talked about the degranulation that took place, and the degranulation took place due to many stimuli, due to C3A slash C5A microbes, etc., etc. And the first hallmark effect is vasodilation, and the second hallmark effect is increased permeability. So we'll come back to that if, as necessary. So initially, let's see what's the best way to approach this. Um, so it wasn't as useful as I thought it was going to be, this flow chart. I do still like that I can see the differentiation flow chart, but the main, the best way to, or the most logical way to organize this information, I thought it ended up being in the following way: a comparison between innate immunity versus ADA, which is a shorthand, which is shorthand notation for adaptive immunity. So here we have a quick little summary. So when it comes to innate immunity, it does not possess memory. It detects conserved molecular families, and it detects cell injury or damage. When it comes to adaptive immunity, yes, there is memory involved, and the specificity for antigen is very high, very, very specific. And this is language used in the PDF notes. There exists exquisite specificity for foreign antigens by the antigen receptors. Cool. So here you have this summary. Oh, and furthermore, when it comes to ADA immunity, the more exposure means more enhancement. So here's another chart, another illustration from the notes, giving information about the comparison of innate versus adaptive immunity. Here is another table. I'll probably combine those eventually. This is just how it worked out so far. So um, when it comes to innate immunity, it pertains heavily to the epithelia due to the fact that these are the surfaces that are touching the outside world. So there's heavy exposure to microbes and potentially harmful agents. So that's why there's that heavy emphasis on protecting the epithelia. And the key cells, when it comes to innate um, immunity, are the neutrophils and the macrophages. So let's see if I made a note of that. So it doesn't seem like it. So one thing that I did was take the stuff that's here, in this column, and isolate it in order to make notes related to it. So let's do that for that summary in order to keep a better track of that summary of innate versus adaptive immunity. So here we'll use this. We'll copy all of that. And I'm not sure where there's space, so I'm just going to put it over here right at the beginning. Damn. Okay, let's add a little space. There we go. So let's open back up. Cool. Okay, we're getting there. We're getting there. Okay, so the key players, and again, that's a pretty nice, that's a pretty important summary. So going back here, the key players for innate are neutrophils and macrophages. So where are the neutrophils? So neutrophils and macrophages. So let's go ahead and highlight those with green. That's when you start out. Okay, so neutrophils and macrophages. This is innate. So we'll draw N in large letters. That's innate. And innate, the key players. And then what were the key players for adaptive? Let's take a look. The key players for adaptive are T cells and B cells. So likewise, we'll go up here, B cells and T cells. So we use a pink color. All right. Okay, good. I'm glad I had a chance to go back and do this summary because this is pretty major, very basic, but very essential, I think. So this will be Ada and Ada. Let's change the font there. Ada for adaptive, Ada for adaptive. Okay, cool. So there's a summary. Out of this whole list of leukocytes, when it comes to the T cells and B cells, those are the ones mainly involved in adaptive immunity. And when it comes to innate immunity, it's mostly the neutrophils and the macrophages, which come from monocytes. All right, so getting back to this chart, I do, I would like to reiterate that here exists, what happened? Here is where exists the epicenter of all of the information and how things are tied together. So here you have this the answer to this self quiz that asks about what is the innate versus the adaptive. Just another comparison. So the first one is what is the molecular basis of specificity or recognition when it comes to innate molecules shared by classes of microbes or damaged cells. And also, this is where the concept of PAMPs versus DAMPs is addressed. And under that same category, what is the molecular basis of specificity or recognition for adaptive immunity? Well, it's virtually any molecule. So, gotcha. Okay, that's pretty important, whether it's the self or the microbe. So, molecular basis of innate is specific molecules, and those are traditionally or evolutionarily or or typically, those kind of words, those molecules are in the on the membranes of microbes or damaged cells. As opposed to adaptive, any molecule can be the basis of specificity or recognition. All right, so now let's address a comparison of receptors. So when it comes to innate immunity, the receptors are germline encoded. They are called TLRs, NLRs, etc. What does that mean? Let's see. Forgot TLRs. TLRs. Let's look it up. TLR. Oh, okay. So this is where that's relevant. So we have to go up, and the thing about receptors are for innate immunity, and notice this is all stuff that stems from the right to the right over here. So, no, sorry, to the left of this diagram or of this summary graph. All of that stuff pertains to innate immunity. So anyway, when it comes to receptors, we have a relationship between PAMPs and the receptors, and there's this variety. There's these different types of receptors. They can be under one of two categories: transmembrane, or they can be cytosolic. And this is where you start to talk about the TLRs versus the RLRs. What did this say? Let's go back to the table. TLRs versus NLRs. So what does that mean? Let's see. TLRs. Here it is. TLRs versus NLRs, which are nod-like receptors. Cool. Cool. So let's see. Phagocytosis. You know what? This is gonna have to get changed and moved around just to fit better, but we'll do that later. For now, no, now's a good time. Let's see. Insert space over here. Come on, what are you doing? Insert space. Here we go. Insert space over here. You're gonna need a lot of space. All right, cool. Very nice, very nice. I'm gonna move all of that junk. Move it to the left. Just for it to make a little bit better sense. There we go. So then we'll move this pink arrow. And this is relevant because this part right here talks about opsonization and phagocytosis. So let's make that connection go that way instead. So this is, I think, how we'll do it. And this will play well with the theme of keeping things on the right. Let's see. To the left, and be able to better avoid confusion. So here we go. Cool. So this arrow is not really going to serve a purpose there. All right, very nice, very nice. That's a nice correction. All right, almost there. Thank you for your patience. All right, so that's where that connection exists. Here, there is phagocytosis being induced via that one particular pathway. Cool. So, and that's that. How does that tie back into over here? Well, it has to do with uh, acute inflammation and such. So, hopefully, sooner rather than later, I'll make that connection. I still don't really see how it's fully relevant, but anyway. So we were here looking at receptors. So when it comes to receptors in regards to adaptive immunity, they are produced. Receptors are produced by somatic gene recombination, and I think this is what takes us into that concept of C CJD CDJ recombination. Let's see. So when it comes to receptors, okay, so this is not it because that's in the notes he specifies at one point that um, there's two different concepts. There's isotype slash class switching, and then there's this other concept. What is it called? It's called, here we go, VDJ recombination. Cool. So how does that tie back into the summary chart? Well, let's see. The receptors are produced by somatic gene recombination. Yeah, there we go. So that's what's happening over here. The receptors for B cells in particular, they are undergoing um, somatic recombination. Yeah, and there you go. There you see the word recombination. Over here, recombine, recombination. So now you have new DNA sequences that are going to code for a distinct um, variable region of the IG and of the TCR. All right, we'll come back to that and make sure we know what that means. But for now, let's keep going with the summary. So let's consider the concept of receptor expression and receptor distribution. So when it comes to innate immunity, there are identical receptors on all cells of the same lineage. However, in adaptive immunity, the there's the key word is clonal. Each lymphocyte has many copies of a single receptor. Okay, moving on to the concept of the basis of self versus non-self discrimination. When it comes to innate immunity, healthy host cells do not have, nor do they display, the ligand. Yeah, okay, yeah. But when it comes to adaptive immunity, lymphocytes with self-receptive receptors are removed from the pool. Okay, so that's a pretty important topic that's being covered there. How to avoid autoimmune diseases. So that's the end of that summary. So again, let's go back here and just review. So these are notes that came from the histology class. And here, this thing, in that class, the professor or professors provided a summary of some details about what are the steps for acute inflammation. So those seem to be kind of not relevant to this course. The notes for this course provided this other summary of the acute inflammation process. It broke it down into three stages. The first out of three is dilation of small blood vessels. 2 out of 3 is the permeability increase or increase of permeability of the microvasculature. And then there is extravasation of leukocytes. Okay, so that's important because um, acute inflammation ties into immune and immune in, so innate. So what type of immunity? The in type, innate immunity. So acute inflammation ties into innate immunity. Likewise, macrophages tie into immune, innate immunity. And does that agree with what we wrote up here? Huh? Where'd it go? Yeah. Yep, macrophages. They are one of the key players in during innate immunity. All right, so the concept, oh, and also neutrophils. Oh, cool. Oh, okay, there we go. That's where the connection, the connection takes place. So neutrophils, they are key players in acute inflammation, and that's why here I drew these arrows, these gray arrows, both converging on innate immunity. And here is a little summary of neutrophils versus macrophages. The neutrophils possess a short lifespan, whereas macrophages possess a long lifespan, etc., etc. So here, phagocytosis gets induced, and yeah, there's phagocyte activation, and then there's killing mechanisms. Cool. So we've addressed that. So let's keep going. It'll become relevant. So, but let's go back to this epicenter. As I mentioned, this is where everything stems from. So, what can we address first? Well, let's just go from top to bottom. So, when it comes to the receptors, well, first up here exists the concept of PAMP and DAMP. So, how does PAMP and DAMP get activated? Well, that gets activated by cytokine production. Here you go. Cool. I'll come back to that with details. Furthermore, what is happening during innate recognition? Well, the PAMPS stands for pathogen-associated molecular patterns, and PAMPS, oh no, I forgot to change this, and DAMPS stands for damage-associated molecular patterns. So the key words or the key concept here is molecular patterns. Cool. So there seems to have been no major details about damps, but except for this, some of the things that should not be located where they are. So also you have, but there is a lot of information about PAMPs, and as we mentioned, there's this variety of receptors to identify pathogen-associated molecular patterns. Those receptors can be transmembrane or cytosolic, and boom, there you go. There you have them. And here is another illustration, but it looks a little bit more realistic, a little closer to a real-life example because it includes a drawing of the plasma membrane, and there is different colors. There are different colors for the extracellular space versus the cytosolic space. All right, so that addresses that. What else? So here is where I saw fit to address the concept of that we covered here. You know, the epithelia. The epithelia is heavily protected. You have epithelial surfaces and potential portals of entry. So here's a summary of how um, the protection gets enacted, what kind of protection exists. We'll come back to that with more details. And here the keyword is multi-layer. There's multi-layer protection. All right, very nice. So, and then here's where it's appropriate to talk about mucus power. Cool. Okay, we'll come back to that if necessary. What is this? ABOD antigen. Mm, that got lost somehow. We'll come back to that if necessary, but that's just a synonym that's kind of important. So we'll put it over here in the corner near the title for easy identification. There we go. Whatever. It's not a major detail. So what else is relevant to the left side of the summary chart? What else? In other words, what else is relevant to innate immunity? Well, you have the complement system. So complement system, what it boils down to is that they are plasma proteins. What's their purpose? Well, they perform an enzymatic cascade. There are two effects of this enzymatic cascade, one and two. There you go. And then here's more details about the enzymatic cascade. And here we go. This is where the connection exists between innate innate immunity and phagocytosis. So there's something about alternative pathway and lectin pathway, classical pathway, and everything seems to converge on this C3 molecule for some reason. And here you go. Here's these details. We'll come back to that as soon as possible. So what's pending? So it might be or might not be necessary to redraw all of this by hand in order to learn it by in order to memorize it. So we'll come back to that if necessary. So here are more notes related to the complement system. How does this pathway get activated? Well, when it comes to the alternative pathway, it's activated by something called tick over. The lectin pathway and classical pathways, it says that they have similar activation, but I haven't jotted down those details. We'll come back to that. So when it comes to the regulatory factors of complement, there are three, as seen here: regulatory proteins, prevention of accumulation, and microbes generally lack some factors. Okay, we'll see what that means later. I'm not sure. Anyway, that addresses pretty much everything related to the left side of this summary chart. And let's go and address things to the right. So here the details are pretty limited. The key players for ADA or adaptive immunity are T cells and B cells. So the B cells, there's a concept called, and by the way, when I write A-gen, that means anti-gen. The A is for anti and then gen for gen, versus A-bod, which is A again for anti and then bod for body. So here we have the concept of A-gen specific clone. That clone will expand / proliferate, and it will secrete A-bods. Cool. So what are okay? So there's this red line going out, and it leads us to analyze these four main features of the ADA immunity, adaptive immunity. It is specific. It has adaptability. It discriminates, and it has memory. So where should we go from here? Okay, so right next door, this is just a divider between those previous nodes and that list of cells that I mentioned at one point. The more the successfully differentiated type of cells. So what's going on here then? So again, the emphasis for um, adaptive immunity as seen in this summary over here. Let's go back and review what we wrote. So when it comes to adaptive immunity, T cells and B cells are the key players. Let's go confirm. Yeah, those two are the key players, T cells and B cells. However, there is that overlap, and that's seen in terms of the APCs, which are the antigen presenting cells. So here I've highlighted the APCs in yellow. As you can see, they converge over here, and the APCs have a lot of involvement in a lot of the immune response concepts. And likewise over here, they they are associated with T cells, and they perform all sorts of things. It induces actions and things of that nature. So yeah, that's pretty important. Anyway, um, for now, let's just focus on. Okay, again, let's focus on this list right here. So let's focus actually on some of the smaller notes. So this is a summary from the second PDF from page 58. So there are DNA changes in antigen receptors, and here's a review of three concepts: VDJ recombination, isotype switching, and somatic hypermutation. So what's going on then? Yeah, we'll come back to that and make sense of it as soon as possible. So here's a small section that pertains to T cells, in particular a subtype of T cell called T helper cell, and it performs isotype or class switching. This part I definitely don't understand too well at the moment, but here is this information for uh, for your for your for your notes. And here's this isotype summary. Yeah, I'm definitely rusty on that. I'll have to invest time into understanding this switching. And that has to do with the spleen and the germinal center. Quite a lot of review. Whatever. For now, let's focus on convergence of the APCs and the effects that they have on B cells. So here you have deep details about B cells, and in particular, the B cell receptor. Cool. So later we will compare that to down here. As you can see, here we have the TCR, aka the T cell receptor. So what's going on? Well, let's go to the short topic up here. There is stuff to know about variable regions. I don't know. We'll come back to that. There is stuff to know about antibody structure. So this is essential. I think this is a more logical way of approaching the material. First, we'll antibodies, examine antibodies in a standalone manner. So they have three types of regions. They have a variable region, they have a constant region, and they have an FC region, which stands for fragment crystallization region. All right. So focusing on the variable region, this is the part that binds to the agent, the part that binds to the antigen. So a lot of diversity is essential there. So how does this diversity get generated? It happens through a process called VDJ recombination, and this brings us back to this summary table over here, VDJ recombination. So we'll come back to that and make sense of it after some more overview of the topics. So yeah, it has to do with loci or loci, da 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 da, recombination, etc., etc. This one was also kind of confusing or challenging, so I'll be sure to come back to that. So that's it for that. Um, then we address finally, we've covered enough information about the attributes or aspects or characteristics of antibodies. Now is when we get into the antibody response, which is pretty complex from the look of it. So here you have a reminder that B cells are the producers of antibodies, okay? And there's two types of responses. You have primary and secondary. The secondary is just more enhanced. In general, you see all these keywords, higher and higher right here, and also shorter lag time. So here is this comparison that I have so far between a primary immune response and a secondary immune response. And again, this is primary um, adaptive immune response, and then secondary adaptive immune response. So here is this overview that the professor provided, and he specified that, hey, if you're able to know what's happening in this graph or in this figure, this drawing, then you know a lot. But he said it's fine. We'll address that in a little more detail later. So for now, it's no big deal. So some things to point out: the naive cells are important because they are, I guess, the starting point, and they're definitely unique or just important for some reason. So um, also here's this different way of looking at the primary versus secondary responses. There's this thing about the peak. I think that the most important thing is this concept of the peak response. As you can see, for the primary response, the peak is smaller, and then for the secondary response, the peak is larger. All right, so that addresses that, and then here's where plasma cells get a little bit of attention, and it has to do with memory and things like that, I think. All right, so let's keep going. Okay, that's it. We addressed everything pretty much related to B cells. So what's going on with T cells? Well, again, all of the antigen presenting cells, they converge and they perform action on the T cells. So before doing that, let's just review some T cell stuff. So there previously was the BCR, aka B cell receptor. Well, here you have the TCR, the T cell receptor. One of the most important things is that this receptor is never secreted, so it's not like the T cells don't make antibodies. Here's where you start talking about MHC molecules, and that's part of what helps to show, display or present the peptide or the antigen. So we address that. Here's the structure or whatever. Here, okay, cool. This is a nice summary from the look of it of T cell receptors, TCRs. versus BCRs. Antibodies like do get secreted, as noted here, versus not getting secreted, they just remain cell bound on the membrane of T cells. So there's that information, and then there's T cell subsets, CD4 versus CD8. There you go. There you go. Some information. And here is this table about subsets of T cells. There are T helper cells, T toxic, or better yet, known as cytotoxic. I just write it as tox for toxic, and then T regular, not regular, regulatory. The T helper contains possesses CD4. T tox, the toxic possesses CD8. We won't address details about the regulatory proteins, regulatory T cells for now. So here are some notes. Different number of functions, there and there. All right. What else? So yeah, now we can address things that are related to the APCs, the antigen presenting cells. So they interact with T cells, and let's go to this first arrow down here. What happens is, so there's something special about dendritic cells. They are the only ones that can activate naive T cells. Cool. Moving on. All other types of cell activation, all other T cell activations that require two things. Requirement number one is that the TCR, the T cell receptor, binds to the peptide slash MHC, and then the second requirement is that co-stimulation takes place. Co-stimulation with the B7 molecule or co-stimulation with cytokines. Here you have information about dendritic cells initiating T cell responses. Cool, very nice. And then the conversation is complicated, whatever. Um, Over here, APCs can be activated or matured. APCs can be activated or matured. Yeah, this seems important, but no time. There's no time now. Okay, so now definitely this topic is very relevant. As you can see, it's called antigen presentation. So um, that's the function of antigen presenting cells. So there's something about currency. There you go. Here's this diagram of how things happen. And there's there's MHC class 1 on the bottom and class 2 on the top. Uh, Class 2 is related to CD4 T cells, and class 1 is related to CD8 T cells. Here's a nice summary of what's going on between type 1 and type 2 MHC. And then here's this thing about genes. This one it really seems important, but also run out of time. You have polygenic versus polymorphic concepts. Not many details about polymorphic, but there are a lot of figures. And somehow that ties into the HIV progression or whatever. And here's this clarification about DNA clarification. That we'll definitely want to address. That's that last video, the one that was only 4 minutes long. Doesn't seem too critical for now. And this is let's spend time instead over here talking about co-receptors of CD4 and CD8. And the main takeaway here is that they do not bind. They are essential for signaling in T cell receptors, but they do not bind. And it looks like that's everything. Yeah, that's about it. Let's just put a box around. Let's draw a circle around this concept so that you don't get confused. So we use that color. We use a pen. All right, there we go. Now things are confined and we know not to mix things up. So all of that stuff in blue pertains to T cells. Very nice, very nice. And that's the end.